concept by the coordinators themselves. So Raymond Schiffelers for Be Smart, Daniele Catalucci for Cupido, Nora Ventosa for Smart for Fabry, and myself for New Deal. So go ahead, please, Raymond, to introduce the Be Smart concept.
Okay, thank you very much, Fabrice. Thank you especially for bringing this all together. You're
welcome.
Let's see, presentation mode, and then we're good to go. Yes. So Be Smart, it's an acronym standing for Brain-Specific, Modular, and Active RNA Therapeutics. And so there are several key words there. The first one is the brain-specific part. We focus on two diseases. One is Alzheimer, of course, a disease with a high prevalence and a high socioeconomic burden. But at the same time, we also focus on spinal and bulbar muscular atrophy, which is a rare disease exclusively present in boys. It's also a neurodegenerative disease, but it has some peripheral components in there, so that might make it easier to address the underlying issues in this case, because the peripheral neurons are also involved in this disease, and they might be more accessible than the central part of the nervous system. As I said in the title, RNA therapeutics, that's what we're aiming for. We are especially focused on siRNA, so they're indicated over here. There's, of course, a multitude of different ways in which you can interfere in this in diseases using RNA therapeutics. But with these, we have two key molecules, beta secretase and androgen receptor, which could constitute possible targets. And of course, they can be adapted by simply changing the siRNA sequence. And as Dr. Boyd just mentioned, siRNA notoriously difficult to investigate, also from a preclinical toxicology point of view, because we're studying a human active ingredient in the surroundings of an animal genome. So the adverse effects might actually be a completely different picture than what you're actually observing in the clinical situation. So that's an additional translational barrier. As I said, the central nervous system, that is one of the most notoriously difficult things to target. Everybody's well aware of all the CNS barriers that exist. Most well known is, I think, the blood-brain barrier. But what we're focusing on, and I think that is an attractive backdoor into the brain, that's the blood cerebrospinal fluid barrier. And one of our partners, Roosmarijn van den Broeke from the Flemish Institute of Biotechnology, is really an expert on this. And she has developed nanobodies. Nanobodies are the small parts, the variable parts of heavy chain only antibodies that are found in the llama-like species like alpacas. And she has identified nanobodies that specifically bind to the folate receptor alpha. And that particular folate receptor is highly expressed on the cerebrospinal fluid barrier. And as a result, things that you attach to the nanobody that binds to the folate receptor can actually transfer into the central nervous system. We also talked about modular. That's the other part in the title or the acronym of the project. We have different types of nanoparticles to which we can attach the different modules. So we have the siRNA on the inside, we have the particles surrounding it, and finally we have the nanobody attached on the surface that would actually bring it into the target tissue. We have the established carriers. Ruth already talked about the Onpattro formulation. So that is the lipid nanoparticle that is really at the forefront of our investigations. We have a couple of polymer-based particles that are following up on that. And then we have the exploratory extracellular vesicles, exosomes, that are constituting possible targets for the future. And of course, their function is to protect and complex the siRNA. Finally, the issue of manufacture, of course, extremely important for these types of nanoparticles. And we do that by microfluidic manufacture. The idea is that the siRNA complexes, while we are mixing the siRNA and the lipids in such a way that the ionizable lipids complex first with the siRNA and form a core to which all the other lipids adhere, and finally the polyethylene glycol coated lipid ends up and forms the corona of the particle. And well, these are the scalable, reproducible ways by which you can manufacture these types of nanoparticles. These are the project partners. I took the picture from Cordis, but every partner is a couple of 100 kilometers off. So these are the partners from the Netherlands, UMCU Utrecht, that's my institute, and 20med Therapeutics. From Spain, University Santiago de Compostela, Flemish Institute of Biotechnology from Belgium, Malvern Analytical and Oxford from the UK, Cinta from Norway, Ibi Lorenzini from Italy, and Loredana will present on behalf of them, and Juris is the project management partner. These are the partners, 3 universities, 2 research institutes, and 4 enterprises. Majority of PIs is actually female, and we are funded in the same call as Fabrice is. So with that, Be Smart is introduced, and I hand over to the next consortium.
Thank you, Raymond. I'll let Daniele for the Cupido project.
Yeah,
Okay, thank
sorry.
you, Fabrice.
I have to stop sharing. Yeah.
Yeah, indeed.
Yes.
Thank you.
Okay. So thanks, Fabrice, for the introduction, and it's gonna be my turn for introducing our project that is called Cupido. It stands for Cardio Ultra Efficient Nanoparticle for Inhalation of Drug Products. So I would like to start my presentation with these pictures because they remind me of my childhood when I was 6 years old, and while playing within this field, I discovered my respiratory allergy. By the time I also learned by the simple use of an inhalation device like this one, I could go back to the field and play again with my friends. So today at Cupido, we believe that a similar inhalation device, but loaded with a nanoformulation, therapeutic nanoformulation, we could provide a therapeutic system for the treatment of a chronic, long-lasting heart failure condition, which is affecting 14 million patients worldwide, having problem with the contractile property of the heart. So our solution is an inhalable nanoparticle. Actually, it's a macroparticle embedding nanoparticle. They are loaded with a first-in-class therapeutic peptide. So when this formulation is inhaled and able to reach the deep lung of the heart, can cross the pulmonary barrier and fast reach the heart, where the nanoparticle can cross the barrier or the cardiac cells, and within the cells release the therapeutic peptide that in this disease condition is able to restore the myocardial contractility and rhythmicity of the heart. So our system that we've been generating in Cupido provide a rapid onset of action. It's not invasive, simple to use, as I was when I was a kid, 6 years old. It's selective. And most importantly, is able to allow to reduce the dose of administration that contribute to reduce the collateral side effect to other organs. And it's able to overcome the issue that you might have with the poor control over delivery from other administration routes. That could be the oral administration, where you have the first pass metabolism, degradation of the gastrointestinal tract, or even the IV administration, which is an invasive. So to reach this system, it was possible thanks to the collaboration of a variety of partners. There are 12 partners that belong to academia, SME, industry, and pharmaceutical compartment. And we can see here from CNR, Charité from Berlin, Simula from Norway, BioMTech from Greece, Sanofi from France, Imperial College in London, Nemera in France, Plumstar in Italy, Finceramica, and we'll have Claudio De Luca talking later in the development process from Italy, in CITC and Life Technology from Italy. So as you can see here is the funding of our Cupido project. And with this, I thank you for your attention for my brief description of the project.
Thank you, Daniel. Nora, we will let you introduce Smart for Fabry.
Thank you, Fabrice. I am going to share the screen. Do you see my screen?
Yes.
Okay, thank you. Thank you very much, Fabrice, and thank you to the organizers to New Deal Project and also the ETP of nanomedicine for the organization of this wonderful workshop. So I will try in 5 minutes to summarize a little bit the concept behind the Smart for Fabry project. Smart for Fabry is the smart multifunctional GLA nanoformulation for Fabry disease. What is Fabry disease? Fabry disease is a lysosomal storage disorder, and this disease is caused by the lack of an enzyme, that is the alpha-galactosidase enzyme. And patients suffering for this disease, they have an undesired accumulation of glycosphingolipids in the lysosomes of cells. And without treatment, the patients die before 45 years. So the organs more affected for this disease are kidneys and heart, and also in some cases, brain. Oops. Attention, because now. So although Fabry disease is a rare disease, and it's a rare disease of the group of lysosomal storage disorders, as a group, lysosomal storage disorders have a great incidence, and still they have and lack a definitive cure. So these represent a serious global health problem. So currently, the most usually treatment for curing this type of disease and also of Fabry is enzyme replacement therapy. So this type of treatment consists on delivering exogenously the enzyme that is lacking to the patients. And in some cases, like for example in Fabry, by intravenous infusion. These, for example, in many cases, the enzyme, like in the case of alpha-galactosidase, this is an hydrophilic enzyme. So this means that the entrance to the cell is very low. And also when it is straightforward delivered to the body, it can be rapidly degraded. It is rapidly degraded. It has a poor penetration on the endothelial cells, and also induces high immunogenicity. So this means that the efficacy of this type of treatments is lower than desired, very much lower than desired, and large doses need to be administered with expensive treatments. So in the Smart for Fabry project, we have used the strategy to achieve more effective enzymatic replacement treatments. We have used the strategy of encapsulating, of integrating this enzyme on liposomes that are also functionalized with targeting peptides. So in that case, as we have seen previously, liposomes are one of the strategies highly or mostly used for the bringing of nanomedicines to the market. But still there are many challenges, mainly related to the manufacturing, because it is very difficult to control the molecular self-assembling of the different components of these liposomes. So in the Smart for Fabry project, we use an innovative manufacturing process that in a one step, we achieve the liposomal product. And more details on that will be given by Alba Cordova from Nanomold Technologies after. So our project was based on some preliminary results at level of proof of concept before the starting of Smart for Fabry. But during Smart for Fabry, we have proved that our liposomal formulation has a larger circulation half-life, also by tuning and by changing the functionalization with these peptides on the surface of the liposome, we can actively tune the enzyme biodistribution, and we achieve a higher efficacy. So our project that is arriving to its end now has been able to arrive successfully to the entrance to the clinical regulatory. And this was mainly thanks of continuous communication with the European Medicine Agency that we have started since the very beginning of the project, which is, in my opinion, the key of our success. And in my opinion, the key of our success is that the team of partners that we have worked together to achieve those results. This team is composed by a research network. I belong to that research network, a Spanish research network devoted to the development of nanomedicines and biomaterials. Then two highly experienced groups in the characterization of nanostructured products. This is the team of Professor Daganit Danino at Technion, doing cryotemp, and also Professor Jan Pedersen Aarhus, university expert in SACS characterization. Then the team of Thomas Brimburger from Joanneum Research, that they are expert in the evaluation of blood-brain barrier crossing. BioCaralty, a pharmaceutical and a healthcare company interested on the exploitation of biomolecule-based nanoformulations, and early adopter, also to make and to help the development of nanopharmaceutics until a bigger pharma can be interested on that. Then Nanomol Technologies is an SME, and it's specialized on the development and scale-up of nanomedicines manufacturing platforms. BNN is a research network and has contributed with the risk and safety analysis, not only of the product, but also of the process for the preparation of this innovative nanomedicine. DDR are specialists on regulation and of medicinal products, and they have helped and guided us in the contact with the European Medicine Agency, and they play a very crucial role in our project. Covance, it's a large company that performs regulatory preclinical testing. Now they are currently performing the toxicological test of our nanoformulation. And Lian Bio also was the one in charge of producing the active protein that was encapsulated inside the liposome. So this
very much, Nora.
A very brief summary, and thank you, and looking forward to the discussion later.
Thank you, Nora.
Thank you.
So I will try to share my screen. That one. Can you see it?
Yes.
So I will be very brief. We can later enter more in details about the strategy combining nanoparticles. We have several nanoparticles in the sister project, but also several active ingredient, peptide, protein, and nucleic acid. So for New Deal, it's the name of the project. New Deal is New siRNA Nanotherapy for Inflammatory Bowel Disease, targeting Janus kinase. So inflammatory bowel disease, they are not rare. They are the most common immune disorders in Europe. More particularly, this disease affect young people, adolescent or young adult, for the onset of the disease. This is related to several inflammation at several parts of the intestine. Let's say also that the patient suffer from several pain, diarrhea, fatigue, but also other complications impacting their quality of life. The current therapy, including corticotherapy and antibody therapy, targeting, for example, TNF, have several drawbacks, and we need several improvements in terms of efficacy and convenience. Just for your information, recently, a novel molecular target has been identified in recent clinical trials, Janus kinase 1 in particularly, and inhibiting this protein involved in the signaling cascade of various cytokines seems to be really promising. So the next slide is just to explain you the concept of the nanotherapy in New Deal. So it's a multi-targeted approach at several levels. At the levels of the tissue, because we target the colon, the inflamed gut, and we have several challenges to overcome to cross the biological barriers, the mucus, the epithelium, but also the plasma membrane of the target cells. It's also a matter of cellular targeting, because we target more especially human cells to inhibit the protein. And the last level, it's the molecular targeting, because this is very specific, on the contrary to the chemical compound, where we target through siRNA, the messenger of Janus kinase 1, and we don't regulate the protein expression, and we inhibit the signaling cascade of pro-inflammatory cytokines. So it's just to give you an overview of the project. In terms of a few key numbers, it's quite the same of other projects. So this involves a lot of partners in France, Spain, UK, Germany, and Belgium. And this is a picture of the team, because it's also a matter of team. I do agree with you, Nora. And we have a great team in the project. So you can find further information on the website of New Deal. So I will stop there. I will not enter more in details, but of course, all the experience of the sister project during the panel discussions. So the first panel discussion that we will open now is the panel discussion of clinical development plan, because I guess that we have nice proof of concept in all the projects. So now our next concern is to promote the future clinical translation. And I let Alan to lead this panel discussion. Thank you, Alan.
Thank you. Thank you, Fabrice.
I will stop sharing.